Hi, everyone. Thank you so much for joining us this evening in celebration of World Oceans Day. My name is Adila, and it's my pleasure to welcome all of you to our event, Our Oceans, Our Life. Now, in this online event, we have three marine scientists who are actively involved in coral reef rehabilitation in Malaysia, and we'll have them on this event to discuss the science that we need to reverse coral reef degradation. Now, tonight's event is not a webinar per se. We intend this to be a sharing session. To that end, we would very much like to encourage everyone in the audience to interact with us on the Facebook Live or on YouTube Live, whichever platform you're on watching us right now. So in the first half an hour, we will have the speakers share with us about their respective projects, and then we will have a question and answer session for 45 minutes. So there's plenty of opportunity for you to ask the speakers any burning questions you might have about coral reef rehabilitation projects. Okay, so if you have any questions or comments for the speakers, please type it down in the comments section below the live video, whether you're on Facebook Live or YouTube Live. You can do it at any given time during the talks. The moderators will pull the questions for the speakers to be answered during the question and answer session. Okay, so to kick off the event, I would like to invite Professor Dr. Sumiani Yusof, the director of the Institute of Ocean and Earth Sciences in University Malaya, who are organizing this event with us, to give her welcoming remarks. Prof. Sumiani, the floor is yours.
Thank you so much, Dr. Noradila, for that introduction and also for starting the webinar. And I would like to thank all of you for joining this very important and interesting session. First of all, Assalamualaikum warahmatullahi wabarakatuh and a very good evening. I would like to take this opportunity to welcome everyone online here to our Institute of Ocean and Earth Science, IOES, Pulley webinar to celebrate this year's World Ocean Day 2021. And on behalf of the Institute of Ocean and Earth Science, University Malaya, I would like to thank the team of Project Pulley. You'll hear about it, very interesting project headed by one of our members from IUS, JFND, for co-organizing this webinar. And this webinar is, as you know, is organized in conjunction with the United Nations UN Decade of Ocean Science for Sustainable Development 2021 to 2030, and the United Nations UN Decade on Ecosystem Restoration 2021-2030, and last but not least, the United Nations Sustainable Development Goals, especially the SDG 14, which is related to life below water. Yeah? And I would like to really express my gratitude to the three panel speakers, people who are very, very passionate about their job and who are very into the restoration and also conservation of coral reefs. They are Mr. Muhammad Ali Said Hussein from University Malaysia Sabah, Mr. Sebastian Zeredi from Coral Q NGO, and of course our own Mr. Effendi Yang Amri from IUS and heading the project Pulley, for sharing their expertise and knowledge in this webinar. And it is also my utmost hope that we consider how we as individuals and as a society as a whole can play a role in supporting efforts to reverse the cycle of decline in the health of coral reefs. And I thank them, especially the speakers, for sharing their expertise and knowledge in this webinar. Hopefully, together we can create more improved conditions and management for sustainable development and of our coral reefs and the ocean. I hope all the participants will find the webinar enjoyable and the discussions interesting and stimulating. Thank you so much and all the best.
Thank you so much, Prof. Sumiani. We are very grateful for the support from IUAS in organizing this event with us. Next, I would like to invite the fearless leader of Project Pulley, Dr. Jillian Uy, to say a few words on behalf of all of us in Project Pulley and also to introduce the first speaker for our sharing session today. Jillian, on to you.
Thank you, Adila. Hello, everyone. Let me start by saying Happy World Oceans Day. We pay tribute to the ocean today, but really, the ocean is not in great shape. And as a result, we have seen an upsurge of coral rehab projects here in Malaysia in the past decade. Now everyone wants to plant coral reefs. However, coral rehab is risky, it is expensive, it is time consuming. And therefore, in every coral rehab project that we do, we owe it to you to make sure that our science is sound, our methods solid. Having good intentions alone is not enough. So therefore, it is really my pleasure to have with us today 3 speakers who have different approaches to coral rehab, but who all want the same thing: to produce the science we need to reverse coral reef degradation, and to do so in a way that is transparent and repeatable. So let me introduce the first speaker to you, Sebastian Zarade. Sebastian has been working on coral restoration projects since 2015, first in the Maldives and now here in Malaysia. He is a member of the Coral Coup Project, which is an NGO that is actively restoring coral reefs and building capacity for restoration in Pulau Lang Tengah. Over to you, Sebastian. You have 10 minutes.
Good evening, Dr. Gillian. Thank you for the introduction. I will start right into my presentation. Okay, good evening, everyone. Welcome to World Oceans Day, as we are going to be speaking today about coral restoration in Malaysia. Thank you for the organizing team for inviting me to the event. Apparently, I'm having troubles connecting.
Sorry, everyone, we'll give Seb a little bit of time. He's broadcasting from Pulau Lang Tengah. As you know, the connection there is not the best. And I guess it's gone off at a very unfortunate time. So let's give him a minute or two.
Okay, good evening, everybody. I hope the connection is stable now, and I hope the presentation is visible. And I'm getting the okay sign. Okay, well, my apologies for the delay in connections. We are in a remote location, so sometimes that can be tricky. Let me start again. Thank you for the introduction, Dr. Gillian, and thank you to the organizing team for inviting me to speak during this event. I'm going to speak about coral trees and clips. What are the techniques for scalable coral reef restoration here in Terengganu, which is in northeastern Peninsula Malaysia. Before I will jump into my presentation, I just would like to introduce who we are. We are Coral Coup. We are a group of Malaysian as well as international scientists, together with stakeholders who work in the area, namely resorts and reef managers. And we have come together to conduct coral restoration. And as the webinar already indicated, what is the research that we need to conduct coral reef restoration? So therefore, we have been doing a long period of research and development to try to understand what are the best techniques that we can apply here in Langtenga. And this year, we have also started to focus more on training and capacity building. Earlier this year, we had the opportunity to team up with our neighbors, Langtenga Turtlewatch, a sea turtle conservation NGO, and we were able to provide training and techniques for restoration practices. And we also really would like to focus more on inviting students to our project to be able to share our experiences and to be able to provide them with opportunities to conduct their fieldwork. Now, having said that, it is very important to first ask the question, why is restoration necessary to begin with? Here locally in Langtenga, we have lost about 60% of coral reefs as measured by hard coral cover. So coral cover refers to the substrate that is covered by corals. And on a single reef scale, this decline has actually reached up to 90%. So that is a very dramatic decrease in coral reefs. And this was mainly triggered by a decline in the population of branching corals. So for those who have never visited a coral reef, branching corals are beautiful 3D structures that grow up into the shallowest part of the reef. And unfortunately, what is very important to understand that restoration is only conducted if recruitment, so natural recovery, is not possible. And we have noted that the recruitment of branching corals is actually very limited up to a failing point. So that's why we decided we need to take action that will increase the coral cover. You see my graph on the right-hand side. I'm showing 3 different reef sites in Langtenga. One coral reef, one site is doing particularly good. It retains a high degree of coral cover, whereas the other 2 are already struggling. A reef with 20% coral cover can be considered sometimes, depending on the case, fairly degraded. So if I was to put that on a simplified map, this is how it would look like. To the east, you see the green zone. So those are the healthy reefs. To the west, you see the red zone. Those are the degraded reefs. And to the left and to the right, you see the blue areas. That is where we are focusing and kind of centralizing our efforts on. And equally important, after asking the question, what and why do we need to conduct coral restoration, is to say what we want to achieve. So what are our goals? Our goals here in Langtenga is to double the live hard coral cover by 2024. So that would mean that the blue areas were those reefs with 20%. So we would like to double that to 40% and to boost the reef structure and function. So boosting the reef structure and function also comes with economic benefits. The reef structure provides the aesthetical value here in Langtenga, which is very important for the tourism industry, but also a complex reef structure triggers the function of the reef, which produces healthy fish stocks, which is obviously very important for the fishing industry. So scientifically speaking, you see the red circle. This is where we are. We are at 20% coral cover, which sums up to about 1,100 colonies per 100 square meter, or 11 colonies per square meter. So we want to double that, and I'm talking now about density simply because hard coral cover cannot readily tell us how many corals we need to plant into the reef to reach our goals. So if we want to get to the green circle by 2024, we need to double the amount of corals precisely to 2,200 coral colonies per 100 square meters. And this is really a step-by-step process. It is not as straightforward as arriving on reef site and just going out into the reef saying like, hey, I want to plant corals. Let's just go out and do it. No, we have done a very long research and development process, which started with baseline research. So think about literally everything from diversity to abundance, to how climate change is impacting coral reefs, what the local stresses are. And then we advanced to preparing our site. So site preparation here means we removed overly abundant predators, such as crown of thorns, which is fairly common in the Indo-Pacific, but is blossoming sometimes due to human factors. In Langtenga in particular, we are having troubles with coral-eating snails called Drupella. And it also means to engage the stakeholders, to engage the community, and to make them understand what is happening and how we can improve it. Then we moved on to nursing coral fragments, opportunity fragments, which are broken pieces of corals on the ground, and to conduct specific scientific studies to understand what we can actually achieve. And today we are in our outplanting R&D phase. So currently we are testing particular methods to scale up our efforts to reach our 40% goal. And only after that, we are really going to reach our last and final step, which is going full steam ahead and planting as many corals as possible back into the reef. And here are the coral trees. So coral trees are really a system that we set up because we need to grow corals to a bigger size, but also it is kind of meant to be a systematic approach, almost close to agriculture. We select particular species. Here you see a couple of branching corals that we systematically and purposefully grow in our coral trees to be able to outplant these later on back into the actual reef. And these coral trees, they have a couple of advantages. They are very wave-resistant structures, and you can see the corals are just dangling off the tree. It allows us to quickly take them off the tree and transport them back to the restoration area. And these trees are also a very good opportunity for us to study what the return on our restoration effort is. So my talk is about scaling up restoration efforts, which basically means to understand what we can harvest in terms of restoration performance of particular species. So in my photographs, you see 3 different species of branching corals that all branch in different ways. And we would like to understand how these functionally similar species differentiate in terms of how they perform. And this is something also that needs to be asked on regional level. So for instance, if we were to compare our results with those from Johor or from Sabah or other parts of the coral reef world, this would really inform us how coral reefs are responding not just to global stresses, but also how they're facing in terms of local stresses. Because local stresses are often very different from site to site and they're very dynamic. So really understanding how species perform can help us sort of create a roadmap that managers can use more readily and apply for their own restoration projects. So our mission, we want to plant 6,000 branching corals per site in the next coming years. And 2 ways to do that would be to the left, epoxy plus nail, and to the right, coral clips. So these are the techniques that we can use to actually plant corals back into the reef, and they're fairly different. So there's a lot of methods out there. Every project needs to accurately identify which is the most suitable methods, and we have tested a lot of these methods, and there are now coming results for our project that let us know how we can reach the desired scale of coral restoration. We are going with the coral clips. In this photograph, you can see 3 pieces of coral fragments that are planted on natural substrate. And at this moment, I would like to emphasize the fact that we are prioritizing natural substrate over using other methods that commonly use artificial materials as the substrate onto which coral fragments are planted on. So you can see that perfectly in these 3 photographs. So this is also our outplanting R&D, and this really reduces the cost, it reduces the dive time, it reduces the amount of foreign material, and it also reduces the training time. And lastly, most importantly, also it increases the efficiency of how we can operate. Because please keep in mind that coral restoration is very resource intensive. It is often associated with a lot of cost, with a lot of time spent doing coral restoration, and often projects are limited in funds and resources. But it's not just that. It also means that once we degrade coral reefs, it is very hard to build them back up, to even just get a little bit close to what they used to be. And therefore, for us to reach 40%, these reefs were 60%, maybe even 80% once upon a time, we need to really identify what techniques can be applied on greater scale. And here you can see that very perfectly. It's bare substrate. The substrate is hardly covered by any other coral. Only left to the tag, you can see a encrusting coral that is starting to grow. And you can see this is perfect substrate actually for corals to grow naturally. But because of the recruitment failure that I've mentioned, that is simply not happening. And that was really the sign that told us that we need to go ahead and plant corals actively to get back the ecosystem function, to get back the structure of the reef. And again, we really would like to prioritize to use all the natural substrate that is available. So this is, for instance, rock, but that can also vary what natural substrate can be, because that really preserves also the natural appearance of the reefs, and it is actually also less resource intense. So I have 4 takeaway messages from us tonight. First of all, we would like to hear from you. If you are a student currently looking for a place to conduct fieldwork, we might be able to help. If you are a researcher out there, especially here in Malaysia, and you would like to collaborate, you would like to share your results with us, we would like to hear from you. And also, if you're about to start your own restoration project, you need some guidance, you need some advice, we would like to hear from you. It is very important that projects and NGOs, governments, and other stakeholders work together as to be able to scale up, because the scale of degradation is really quite high. Please define your restoration goals, the resources that you need, report on your failures and also on your successes, because this allows other people to learn from your work. And it is a constant learning by doing and sharing, and that really enables us here in Langtenga too to increase and improve our methods. Lastly, I said it a few times already, work with the natural reef substrate before adding any artificial materials. And the last message, I prepared the last slide just for the last takeaway message, which is we have to cut global greenhouse gas emissions as we restore coral reefs. You hear this all the time in the news, climate change is going to have devastating impacts on coral reefs, both here in Malaysia and globally. Climate change has been impacting Malaysian coral reefs since 1998. To the right hand, you can see a temperature graph that I've put together for Langtenga only. You can see an exponential rise in sea surface temperature since 1985 until 2020, which means this temperature is able to mass kill corals in a process that is called coral bleaching. But the future is not yet set. Things are not doomed. It is still in our hands to try and mitigate, perhaps even save what is currently left out there, and that is what the left picture is for. You see 4 examples of branching corals particularly that have gained an increased heat tolerance over the time, and this really tells us there's a window of opportunity to act. So if you're listening today and you're wondering how you can get involved, spread the message and demand greater climate change actions from your business leaders, from your local government, and from literally everyone who can help and contribute. Thank you.
Thank you so much for sharing your project, Sebastian. That was a really cool presentation. And it was really cool to hear about scalable and affordable coral nursery systems and also outplanting methods. And it's really wonderful to hear of your work to restore the reefs around Langtenga, which could hopefully serve as a model for other efforts in the future to restore similarly degraded reefs around other islands that have been impacted by tourism. Thank you. And we look forward to more updates from Coral Coup in the future.
Thank you.
Next up, thank you. Next up, we have Mr. Muhammad Ali Said Hussein. He is a marine ecologist and a lecturer with Borneo Marine Research Institute at the University Malaysia Sabah. Mr. Ali will be sharing with us about a coral relocation project in Kota Kinabalu, Sabah, where coral fragments from a deteriorating reef area were relocated to a nearby island in an effort to retain the coral biodiversity of the area. Mr. Ali, anytime you are ready, you have 10 minutes.
Thank you very much, Dr. Noradila. And I'd also like to thank Project Pulley for inviting me to share the little bit of effort that we have done in Kota Kinabalu. Let me just share the, hold on, yeah. Let me just share my screen. Right. Okay. Right. So we conducted a relocation effort from within the Sepangga Bay in Kota Kinabalu, Sabah, in the midst of the current pandemic. So it had to be a quick effort that that was conducted over a short period of time to relocate or move the corals from this particular area in Sepangga Bay. So this coral relocation program was initiated by Sabah Ports in collaboration with UMS as part of the Sabah Ports corporate social responsibility program. Due to the rapid development in the area, in the inner Sepangga Bay, the remaining corals that can be found in that area is undergoing very high threats, including very high turbidity as well as episodes of red tide. Right? A survey of this area showed very low coral cover within the bay, restricted to about 10 to 15%, and this is mostly restricted to very shallow sandy areas. We conducted the relocation exercise during February and March, right at the beginning of the pandemic, and we had to break for the MCO, and we were able to continue again in July of 2020 up to September 2020, and we were assisted by Dreamer Islands Resort and Mari Mari Sepangga, located in Pulau Sepangga. So this is a picture of the bay, the inner bay of Sepangga, and you can see a lot of development, including the port activities in this bay. Across the bay is the naval base, the TLDM naval base, where the Scorpion submarines are located. Right. So a basic background of the Sepangga. It is located approximately 10 there's also an oil terminal and a cement jetty in the area. Recently, a shipyard has been established in the area, and also the naval base. So practically the whole bay is lined by industries, and there's also several villages located along the coast, namely Kuala Megatal, Kampung Numbak, and Kampung Gentisan. Right? And in the surrounding area, urbanization is rapidly increasing, with the presence of Kota Kinabalu Industrial Park and various commercial and residential areas in the surrounding area. UMS is just located next door, further down across the river. Right. So basically what we tried to do was we were trying to remove the remaining corals in the inner Sepangga Bay to a nearby island, Pulau Sepangga, which is located across the bay, approximately about 5 kilometers away. Right? So most of the corals are restricted in a small area within the bay, which you can see in this image as shallow patches. Right? So these are the areas that we targeted. Right? It's located very close to the Sepangga container port as well as the oil terminal. That's why Sabah Ports was interested to collaborate with UMS to hopefully give a better and more suitable location for these corals, instead of letting it degrade over time due to the intense threats that's happening, right? And initially, the area that was discussed covered approximately 900,000 meters square, but as most of the corals are restricted to very shallow areas between 1.5 to 6 meters, these areas were much reduced, and of this area with depth of less than 6 meters, the coral cover was only less than 15% in average, between 5 to 15%. So when we were trying to relocate the corals, among the hazards that we had to face, especially in the inner bay itself, was very high turbidity, whereby as we go deeper, visibility can drop below 1 meter, and beyond 8 meters, we could not do any effort at all, because there was no coral for one, and we could not see anything at all, not even the hands in front of our face. That area is known for jellyfish, right? Lobotomyces that is occurring seasonally, and unfortunately during our relocation, there was a small bloom of Lobotomyces, and also the box, as the area is also very heavily used by the surrounding villages. We had a lot of boat traffic, mainly trawlers and pump boats. Although it's also the location of a port, we were not having much issues with the ships, because where we were working were very shallow areas, and the ships tend to stay away from this area. There were a lot of sharp objects. The corals were sharp. There were urchins, and there were also, unfortunately, various discarded materials such as cans and glass and so on. There were also venomous creatures that we had to keep an eye on. And most importantly were the dives. As we were trying to relocate as quickly as possible, we were conducting 4 dives in a day, 2 dives in the donor side and 2 dives in the relocation side, where we would conduct removal activities in one side through 1 dive, go across the bay to the Pulau Sepangga to relocate the corals, and then return again to retrieve more corals, right? So this is the current. This was the conditions that we were working in. In this case, the water is slightly tinged green due to the algae bloom, right? The transfer of corals were done using plastic crates. We removed the corals from the site, the donor site, using these crates, and we would pile them in a predetermined location in the shallower areas underwater until we have filled all these crates. And only once we have completed filling up all the crates, we quickly bring it up onto the catamaran, the UMS catamaran, which we use. And during the transfer between the inner Sepangga Bay to the island, we sorted and did a quick record of the various corals that we relocated. We opted to transfer the corals without water because of the short interval between the sites, and we could, handle the corals in order to identify and sort the corals quickly. And from past experience, we have also noticed that when we had the corals in tanks with poor water exchange, meaning we don't keep changing the water, right? Then the corals will start producing mucus, and somehow the survival would drop fairly drastically, likely due to the lack of oxygen and maybe some other issues as well related to the mucus that they produce. In total, we managed to relocate approximately 4,000 pieces of corals. Most of it was Goniopora. We had about 1,000 pieces of Goniopora, followed by Fungids and Poraites. Most of these corals were free living. We could just simply pick it up from the substrate. They were located on very muddy substrates. They were not attached to any rocks or any significant hard substrate that would what do you call it? Make it more difficult for us to relocate the corals. They were fairly small in size, but yeah, once we fill up the crates, it can be quite a struggle to bring it up and down the water column. Interestingly, within the bay, we managed to identify 44 different genera of corals in the exercise. And this is an interesting thing, I thought, because a particular postgraduate student, Muhammad Firdaus, found 44 genera in the best sites around Sabah Bay, and a total of 59 genera in the whole Sabah Bay area. However, diversity at species levels is much lower in the inner Sabah Bay due to the very challenging conditions in the area. As it was a rapid relocation exercise, we could not properly tag it, but we were able to cluster the corals in groups of specific genus during the relocation exercise. As you can see here, this whole area is covered by corals of the same genus, right? And we hope to continue monitoring the progress of the corals in the future to see how the corals perform in this new relocated site.
Hi, Mr. Ali.
Yeah.
Just wanted to wrap up.
Yeah, so I'm finishing up. So this is the one of the corals that we moved, which was quite iconic in the sense that it was interestingly shaped like a flower, which was also in the cover. And this photo was taken 6 months. Together with the previous photo, it was taken 6 months post-relocation, and it seems to be doing fairly well. Thank you.
Thank you so much, Mr. Ali, for sharing what sounds to me like a rescue mission, essentially. And I'm sure it was not an easy task to dive in shallow waters in such an area impacted by heavy development, looking for very few last corals remaining while avoiding danger. And the fact that you managed to do 4,000 fragments, that's actually quite a feat, and especially in the time frame that you had access to. We look forward to hearing more updates from your team on the transplanted corals sometime in the future. Thank you so much for your presentation.
Thank you.
Okay, last but certainly not the least, our third speaker for tonight's sharing session is Mr. Effendi Yang Amri. Mr. Effendi will talk about the science of coral rehabilitation by Project Pulley, explaining how the project aims to find ways to improve rehabilitation by looking at it from 3 different aspects. Effendi, the floor is yours for the next 10 minutes.
Okay, thank you. Hopefully everybody can see the slide.
Not yet.
Hang on.
Yep, it's up now.
Okay, so Pulley, thank you very much. So Pulley is short for Protecting Underwater Life Through Integrated Rehabilitation. So I'm going to share the science behind Project Pulley now for the next few minutes. So the problem is actually starting off from the breakage of coral reefs. This is due to climate change or from us humans, our activities. So once the corals break, the ecosystem will collapse. The coral reef ecosystem will collapse. And why is this so important to Malaysians especially? Is because it's our seafood. we're going to lose our seafood supply will be threatened. So in the past few years, people have been looking at this and are doing active intervention using coral rehabilitation. But we now know that most of the time this is failing because survival rates are low, or basically we don't know, it's unknown, it's not reported. So Project Pulley would like to improve this, the solution to this failing solution, by looking at it at 3 scales. So first, we are looking at the colony scale. Second, we'll be looking at the molecular scale. And the third, we'll be looking at it at an ecosystem scale where we'll be using herbivores like sea urchins to improve the coral rehabilitation. So the first scale is colony and its speed. Yeah. So what we do for the first sub-project of the first scale, this is basically mostly done by our postgraduate student, Ms. KL Chu. It's looking at coral recovery rates of 2 different types of coral life adaptive strategies. One is called, basically, is R, which is actually just fast-growing corals, and K, which has the life adaptive strategy, which is basically slow-growing. So for the fast-growing corals, usually is the Acropora species. And this is the one type of coral that is usually used for most of the coral rehabilitation efforts because they grow fast. And you can see that, oh yeah, we're getting a success. But what about the slow-growing corals, which is the K? Is this better to be used? Because for the Acropora corals, usually the survival rate is quite low. But Echinopora, since it's slow-growing, is more robust. So for this project, we are not just looking at the fragments of the coral. When we mean fragments, is actually when you cut the corals, the branching corals, the fragments of the coral, and transport it to the nurseries. That's what we call as fragments. Usually people just look at what happens to the fragment, what is the survival rates and how's the growth rate and whatnot. And people always forget about what about the donor colonies, the ones that you took the fragments from. How are they doing? So that is one of the questions that we wanted to ask in this sub-project. So when we did that, basically, just look at the donor colony first. So remember, the donor colony is where we took the fragments off, we cut the fragments off from. And we can see that the R corals had a survivorship rate about 2% better than the K slow-growing corals. And the average growth rates is about 3.5 times higher for R compared to K, and the lesion recovery rates. Lesion recovery rates is basically where you cut it, how fast does it recover? It was 7 times faster in recovery for the R-type corals. But when you look at the fragments, where that is one that has been transported to the nursery to be grown up, the survivorship of fragments for R was nearly 20% lower compared to the K. But as what had been expected, because they are fast growers, the growth rates is about 1.5 times higher than the K, and the lesion recovery rates was about 5.5 times higher than K, which is expected. Yeah. So when we look at this, you can see that actually the K-type corals, which is the Echinoporas, is actually better at survivorship on the nursery. But we need to look at where the donor coral colonies, where we took it from, it was less than the R. So we need to find out how, for the next step for Pulley is to find out how we can make this better for the donor colonies for the K corals. But this just shows that, yeah, we should and could use other types of corals for our coral rehabilitation. The second scale that we were looking at was at the molecular. We're just looking at age. So when you see a coral colony and you want to sample it for fragments, is there a difference from taking the branch at the bottom or the lower part or the upper part? Is there a difference? So for our work here is that we posit the question is that are the lower branches on a colony we can deem as old versus the upper branches of a colony which we can say that they are younger. So would there be a difference in their recuperation rates and whatnot? So how do we measure actually age in corals? So what we are looking at is actually using telomeres. So telomeres they act as buffers for the ends of our DNA, and here are the telomeres. So they actually shorten with age, and this is what we call as molecular aging. So when it is older and you have shorter telomeres, the organism is actually it can be said that it has a decreased regeneration. So older corals, which is basically what we are thinking is the lower parts of the coral colonies, the branches, if you take them, would have decreased regeneration rates. So if you want to run a coral rehabilitation effort, which one would you choose? So basically, we are trying to estimate age by comparing telomere lengths in upper and lower branches. Okay. During our work for this, we just managed to do some preliminary work because of when we were on site, the pandemic came, and so we didn't have time to get a lot of samples, and this is still in the working stage, and we're still getting more samples for this. However, the group this is actually done by mostly done by our postgrad student Encik Jihad Wajdi. Looking at, so when we looked at growth rate versus R versus K just now, and also by young versus old, this is a graph of Acropora, or basically the R, the fast growers, and this is Echinopora, which is K, and the slow growers. You can see that actually when we looked at the linear extension of the branches, linear extension means it's growing, it's elongating. Yeah. So the variability for the younger corals was much higher than the older corals, but we didn't manage to get significant difference because of low replicate size. So we're hoping to go back again and get a better replicate size and to see, is it really both Echinopora and Acropora showed the same kind of variability for the young branches. Second, we looked at the surface area for the Acropora, which is the R, and this is the Acropora, and which is the K, which is the slower growing corals, is the other way around. Surface area, that means they are not elongating, but they're going fatter, they're getting larger. So the variability is more for the older corals. So there are trends showing that young and older branches are giving different results. And this could be very important in the long run if you're doing coral rehab. The third scale is using ecosystem, which is looking at herbivores. So basically for this, we're looking investigating the role of herbivory. This picture shows a very nice coral, colorful, is growing well, but if it's gone through stress, like a mass bleaching event, or it dies because of sedimentation, there will be what we call as a phase shift to an algal dominated reef. Once it goes into an algal dominated reef, the problem here is that there is no longer a space for new corals to resettle and regrow and make a phase shift back to a normal coral reef. It's very hard because the algae is more dominant. So what the theory is here is that we want to use herbivores, like mostly urchins, to eat away the algae to make space for the corals to come in. Yeah. So this part of Pulley, the first experiment that was done was to look at just by putting down cages. So these cages, basically what it does is one area, when you put a cage, herbivores cannot come in, and therefore we want to see if there's more algae and if there's more coral spat. Coral spat here is actually coral larvae. Are they coming back again? And we have another area which has no cages, and we just want to see if therefore the herbivores can come in and clear the space. The second thing that we wanted to look at for sea urchins is their mitigation patterns and homing behavior. So basically, the corals were tagged. This is an interesting one. If you want to see, you can see it on Project Pulley's YouTube, how we was done by our postgraduate student. This part of the work for Project Pulley is handled by a postgraduate student, Miss Mok Man Ying. So she tagged the urchins and followed them for 72 hours to see their migration patterns and if there's any homing behavior of these urchins. Where do they go on the reef? Where do they eat and whatnot? So our preliminary result from the exclusion cages experiment, one, we could really see that actually if you put on the cage, there's an exclusion cage. This is the algae biomass. The algae biomass is much, much higher compared to the open cage where the herbivores can come in, basically the sea urchins. So sea urchins do eat and clear the algae away. Yeah, and also for coral spats, we could see on the tiles a lot more coral spats. So the tiles were in the cages and outside of the cages to see how much corals could redominate the area or not.
Effendi, sorry to interrupt. You have to wrap up soon.
Yep. Okay. Yep. So for sea urchins, basically there's a home range of 8 meters, and there are patterns of their movement that we are looking at it right now. In summary, for Project Pulley, for Nurin, the donors and the fragments, we can see that the donors R is better, but for the fragment survival, K is better. For the molecular age, we're not sure yet because we didn't have enough replicates. And for ecosystem herbivory, yes, sea urchins can control the algae. And the other part is one urchin, there's an 8-meter radius. So basically, we can use one urchin can actually garden a reef at an 8-meter radius, and that could be important for coral rehabilitation. So for us in Pulley, we are at the first stage of coral rehabilitation experiments, which is at the donor and nursery level. But there's a third space where Coral Crew is doing, which is at the recipient. Where do we want to put to transplant the corals out to? And phase 2 will begin in 1 or 2 years' time. We need to remember that for any coral reef restoration planning, you need to have plans and design. There's 6 steps, and unfortunately, most of the coral reef rehabilitation work in Malaysia does not have all these 6, and this must change. All reef restoration efforts must have all these steps. In Malaysia, we are actually basically, most of us, a lot of the coral restoration efforts is actually just shooting in the dark, or these darts. We need to really choose what type of coral to be used. Can we use reef animals around the herbivores? And basically, the last thing that's really important is we don't have long-term coral monitoring to show that actually all our efforts is a success or not a success. Thank you for your time, and these are the names of my colleagues and the postgrads involved. Thank you very much.
Great, thank you so much, Effendi, for your presentation. I hope our followers have a deeper understanding of Project Pulley's multi-scale approach by now. Okay, so I would like to thank all of the speakers again for sharing about their projects. So now that we have heard about all of these really cool coral rehabilitation projects that are ongoing right now locally, we can kick off the question and answer session. So I would like to invite all of the speakers back onto the stage or the screen. All right. Okay, so I'll start with the first question, and this is for Effendi. What is the difference between restoration rehabilitation, and transplantation?
Thank you for the question. I was expecting this one because luckily you got all three speakers. Each of us had restoration. One had restoration, one had rehabilitation, one had transplantation. Well, the basic difference between restoration is that restoration aims to get back the reef back to its natural state before this. Just get back to what was before, right? That's the big aim. Rehabilitation is to just let it survive. We just make repairs, and the intervention is more for, okay, we want to make sure that it's alive, but it might not return back to the previous stage. So rehabilitation is actually a subset of restoration. Yeah, transplantation is actually to move some corals to a new place. That new place, we don't know if it's for restoration or just to bring it over to another site, because the actual donor site is going to get destroyed by something. So for me, that's the difference between restoration, rehabilitation, and transplantation. For Pulley, we chose rehabilitation because we do not have enough data and knowledge to be able to restore a reef back to its previous state. We hope to get to our restoration levels very soon.
Thanks for your answer, Effendi. And now we'll field the question from Puan Izarena. Puan Izarena is from the Department of Fisheries. She asks Sebastian, when we do coral survey and the result for certain sites shows poor live coral cover, it doesn't automatically mean that the area is degraded, right? It might be natural, due to natural factors, or the environment was not suited for the growth of hard coral. To identify an area as degraded, we need a set of data that shows trends of the live coral cover. Do you agree with that, Sebastian?
I absolutely do agree on that. Coral cover is really just the first measure that people use to describe the state of the reef. And actually, I would even go beyond that. I would say that even a healthy reef would say 60 to 80% coral cover may not actually be healthy, because there may be something going on and it might be in a downward spiral. So what we are not able to present is, of course, all the elements of our research. I would say that I think a lot of people are doing more than just coral cover analysis. Here we also recorded the substrate of coral rubble, which is basically that coral that has already kind of collapsed, and you can see it on the reef. And for those reefs that I've mentioned that are degraded, the extent of coral rubble is sometimes even up to 40%. But that is one indicator that the degradation is not natural, in fact. And there's also a fair amount of sand, which indicates sedimentation. That is something that the other speakers have also mentioned. So yes, I do agree with you. It's a very good question, and there's a lot more research needed than just to talk about coral cover. But coral cover is a metric that people understand quite easily, and it's also one of the more easier things to report.
Thank you so much, Sebastian. So the next question is from a viewer, Marcus Wong. He would like to ask, how do you select the type or species for coral replantation, and how do you plan to maintain the diversity of the species in replantation? I think, Mr. Ali, do you want to take this question?
Okay, yeah. I guess partially it's through experience. You sort of have an idea of what sort of corals survive better in certain conditions. Like this project that I was doing, rehabilitation, you can see that most of the corals were free-living, large polyp corals, and they survived in very muddy conditions, right? So we chose to replant it in a fairly similar area rather than sandy substrates, whereas an area, what do you call it? A different species may survive better in a different location. Acropora, for instance, would do quite poorly in the place that we transplanted the corals. in the leeward area of the Sepangga Bay because of the conditions. And naturally, if you look around the surrounding area, there's very low percentage of acropora corals in that area, and the likelihood is that transplanting a species that is not found in a particular area would be a practice that would be futile.
Thank you, Ali. Let's move on to the next question from Faz. Faz has 3 questions here, but I think question number 2 and number 3 have already been sort of addressed by the speakers in their presentations. So I'd like to ask if there's anyone among the speakers who'd like to answer the first question from Faz. The removal of COT, crown of thorns, through injection of vinegar, is this a valid method? And will it affect the nearby corals? Who would like to answer this? Effendi?
Thank you for the interesting question. Yes, cod removal using injection of vinegar is one of the best ways to do cod COT removal. It is a valid method, and it's been used in a lot of places because it's very easy to use, and it will affect the nearby corals. If the vinegar concentration is not too high and that you're not using a lot of vinegar, it should be fine. And also it just disperses in the water, but mostly it's being absorbed by the COT. It should be okay. For the other 2 questions, what type of corals are easily planted? Branching, massive, tabular, that depends on the area that you're going to put it in. I would say branching is easier compared to massive, because massive, you have to carry it underwater. I don't know how big massive is going to be. It's going to be very, very heavy. Number 3, natural substrates or artificial substrates give better result for growth of planted corals. That one, there's not a lot of data out there for that, but I would always say try to use natural substrates because we don't want to create too much pollution in the water.
May I add on to that regarding the vinegar and the COT? I would also just put into the round that when we do such drastic measures, such as removing a species or like culling a species that is actually quite natural on the reef, you also have to ask yourself what happens if you don't do it. Particularly chronophorans, they're very fast to reproduce sometimes, and once there's an outbreak, they just really go quite manic across the reef and they just eat everything that is in their way, tabular corals, branching corals. And I think that has been an issue in Malaysia and a lot of places. So the question is, if you don't do it, is it going to get worse, or is it just naturally going to die off? And there's a lot of research that says, well, you better do it because crown of thorns can be very destructive if they reach those levels.
Thank you so much for sharing your thoughts, Effendi and Sebastian. Next up, let's hear a question from Mabel Tan. So we've heard about relocations and transplantations in short distances, but what if are you able to relocate corals successfully from, say, Tenggol on the east coast of Peninsula Malaysia to Port Dickson, Blue Lagoon on the west coast of Peninsula Malaysia? Is that something that can be done? Any of the speakers would like to take this question? Perhaps Sebastian?
Well, I'm not. I haven't done something like this before. Langteng is really quite a small location, but just my first thought, my intuition would be, don't. It's a really complicated process in just moving a coral from one side of the reef, 10 minutes by speedboat, and doing something as drastic as moving coral colonies from the east coast towards the west coast is perhaps logistically a great challenge, but also ecologically. The west coast is a different ecozone. There's probably very different conditions going on there, and that requires a lot of probably just baseline research to understand what is really going on in both sides, what is really different among both sides. So I would if I would be in a position to make such a decision, I would be very careful before jumping into a project like that.
Thank you, Sebastian. Mr. Ali,
So-
do you have anything to add?
No, I agree with what Sebastian has to say, and from the experience of translocating corals from the inner bay to the bay of Spanga, the logistics is quite challenging. I would doubt very much that you'll be able to move a lot of corals from Tenggol to Port Dickson due to the various logistics involved. And I wouldn't do it for, similarly due to the eco zones that Sebastian mentioned.
And the next question we have is from Pulley. We'd like to know, is there any guide or instructions available for those resort and dive operators if they're interested to work out their own coral restoration activity? Could I ask Effendi if he would like to answer this?
Yes, there is. In 2020, there was a publication that just came out. It's a manager's guide to coral reef restoration planning and design. It was published by the NOAA Coral Reef Conservation Program, and the first author is Shaver. You should look at that. And
How
also,
do you spell that, Fendi?
Shaver,
SHAVER. SHAVER, Shaver. Thank you.
Yeah, so it's just published in 2020 last year. And also, I would say that some of these manuals could be daunting to read, and maybe you could find someone like a marine biologist, or better yet, a coral biologist, to be brought onto your team, because there's a lot of graduates out there that could help you out to make you transfer the knowledge from the manual to an on-site hands-on situation. I would say yes. Please do get the manual and get a marine biologist or coral biologist to help you.
Yes,
and
can I jump on that? Because
yeah.
Yes, I know. I just agree so much with everything you just said right now, especially on for resorts to bring in actually marine biologists. My first experience in the Maldives was particularly that the resort had established a marine discovery center. They developed programs that actually also engaged the guests of the resort. So that there's a lot of things that resorts can do, and there are guides out there, but definitely having a qualified team on site is something that most resorts should look into. And this is also what is happening to us here. We are being hosted and very much supported by Summer Bay Resort here in Langtenga, and they're very much helping us to get everything going. So yes, definitely guides out there, marine biologists, send them to the ground and get the job done.
Thank you, Effendi and Sebastian. Next, we'll hear a question from Nur Salwa. She asked, what are the indicator animals that can be used to show that a coral replantation has been successful and the corals that have been replanted are healthy? Perhaps Sebastian or Ali would want to address this question? Sebastian, maybe?
Okay, I'm afraid I'm just being too talkative, but what are the indicators that can be used to show that coral replantation is successful and healthy? Fish abundance. I think that most case studies really highlight how coral restoration can impact the structure and complexity of the reef, which is responsible. So for those who maybe not have heard this before, the complexity of the reef is responsible for the habitat for the fish. So there are studies, or the common thing to say is that a quarter of marine life actually at some point during their life cycle lives in coral reefs. So now imagine that coral reefs only cover 0.1% of the ocean floor, and yet they are this incredible oasis of diversity. So if you plant corals into a site that had a low fish abundance and your corals survive, and then you see that the fish abundance is increasing, that may be possibly due to your restoration project. And I think that is one of the most common things that people would look into. I'm pretty sure there's other things as well, but this is something that is commonly addressed and looked into.
Okay, next question. Next question comes from Faiz Hanafia, who has asked, what is your opinion in restoring slow-growing but more resilient coral genera? I suspect that this was in response to Effendi's talk, actually, where you talk about R and K, right, Effendi? So maybe if we could start with Effendi answering this, and any one of the speakers can also jump in after that.
I would say to this question, thank you for the question, Faiz. It's a good question. Yes, my opinion for this is that we should use the slow growing ones, which are more resilient, because like what Sebastian also said, we must remember that the climate change is going to happen and is happening right now. The sea surface temperatures are going up. There'll be more storms. There'll be more intense storms that are more intense. And so we need more resilient coral genera out there on our reefs, and maybe also on our nurseries. Because if we keep on growing the fast growing ones, which is the R, they are actually easily killed by any changes in the environment. So we might lose the whole plot of our rehabilitation effort. So we need to have a good balance for the slow growing ones, and that is more resilient, to be placed not only on the nurseries, but also outplanted into our reefs, to make sure that anything that we do, the reefs are more resilient in the long run. To have that, and also we can get more diversified corals on our reef, rather than just one acropora type corals, and you'll get 100% coral cover, but it's only one species, monospecies, and that is a dangerous thing to be in, because then all will die by one event, compared to have a multitude of species which is more resilient and whatnot. So only a patch dies and not all of them. Yep, I will say yes, more resilient coral genera.
Thank you.
Oh, sorry, you want to go ahead, Jillian?
Go ahead. That's a very interesting question here by Go Yu Hao. And I'm trying to relocate it back again, but I'm wondering if anyone was interested in taking this. And he asked, instead of mutating corals, why don't we try coral crossbreeding? So obviously, this is a question for a geneticist. And if so, either the speakers can jump in and answer it, or if not, Adila, you're a geneticist. You can also help save them by answering this question. So go ahead. Why don't we try crossbreeding?
Okay, I will let the coral ecologist start first, perhaps.
Okay, mutating coral. So if it's humans, it's going to be like X-Men. This is X coral, right? So mutating corals. Instead of mutating coral, why don't we try coral crossbreeding? Yes, there are already efforts in Australia by a group led by Madeleine Van Open, and also in the US, and a group of other coral researchers out there. They are already crossbreeding corals to find which is the best, because they found out that some coral species are better at handling high sea surface temperatures, and some corals don't in an area. Say compared, I'll just take a example, one in the Red Sea and one in Australia. If they can crossbreed the ones from the Red Sea, where it's already adapted to high temperature, and they just crossbreed it and then transplanting it back in Australia and whatever. So they are trying the crossbreeding, but there are dangers in introducing new species to new areas. So I'm not sure how they're going to sort that out, but that's my answer for this. Yes.
If I could add, the other thing that scientists are currently doing, other than actually crossbreeding the coral host, they're also actually trying to I wouldn't use the word mutate, but one of the projects that's being led by Madeline Van Open in Australia is she's using directed evolution. So basically, what her team is doing is they're culturing the microalgae that actually live in the corals, the zooxanthellae, so the ones that actually help the corals survive. So they're culturing the algae in the laboratory and exposing them to higher temperatures so that they can actually direct these algae to, I guess, acquire the genetics to resist higher temperatures. They haven't actually done field experiments, if I'm not mistaken, but it is in the hope that in sometime in the future, if we ever come to a situation where we actually have to go in and manually replace the algae in corals on site with corals that we grow in the lab just to have them survive, that's something that is within the possibility of actually achieving.
I also agree on. Yes, I would love to actually, because I really agree with what Mr. and Dr. Adila just said, but I also would like to put a reminder out there that while this research is truly fascinating and it's incredible that science has come to this point, we also have to remember about the scalability of such interventions and about the resources needed. And also, while this is definitely something that needs more investigation and can become perhaps further down the line an important technology in reef restoration, it needs to be said that this should not be an excuse for drastic climate change actions, because crossbreeding, there are 831 coral species out there at this moment, and we haven't really gotten the full picture. So if we just manage to crossbreed 100 corals to a point where we can maybe plant them on a few square kilometers of the reef, it is nothing in comparison that we could achieve if we really take action on climate change. So great science, fantastic science, very interesting what's coming in the pipeline from various researchers across the planet, but climate change action is first.
Okay, coming up to the next question. This is for Ali from Marcus Go. He says, great job on the coral relocation. My question is, other than the flower arrangement of corals shown, what is the overall survival rate of the rest of the corals?
Okay, thank you, Mr. Marcus. Yeah, as I mentioned earlier, the problem with our effort, it was smack right in the midst of the pandemic, and we were restricted by the permission to go to the field to assess. So the few times that we were able to go to the field to assess the corals has been very brief. In general, my overall impression was that the survival was fairly good. It's not 100% for sure. And there were several species or several genera of corals that did not do very well. But overall, the survival of especially Goniopora, for instance, did very well. There were others such as Tachyphylla that did very poorly, and I suspect that it's due to the location that we placed the corals. It's not even across the board for all the corals, but in general, most of the corals did fairly well.
And since Sia's here, Ali, I just need to flash the next question. Sorry for jumping in like that, but because it's related, a lot of people are really excited about your relocation work because it sounds like Mission Impossible. So Fabian Sukiato asked, how long did this relocation project take, and how many people were involved?
We had because we didn't want to have too many people in the water at the same time because the area was quite turbid, and we had a limited amount of space, and we had to follow a certain SOP. We had to reduce the number of people on the boat and all that. We had about under 10 people, and we did about 60 dives within the time. If I'm not mistaken, 64 dives relocating and placing the corals. Okay.
Less than 10 people sounds like we really needed a lot more people there.
I was very thankful that the percentage of the coral cover was very, very low. It was very low. In most cases, it was under between 5 to 10%. The best place had about 15% coral cover. And unfortunately, we were not able to monitor on a regular basis because of the MCO. We finished relocating in September, and there was the second phase of the MCO where we had to lock down after the Sabah state election, and we couldn't go out to the field again. And we were only able to go back out in March. And now we are back under MCO. So it has been very, sporadic sort of assessing of the coral relocation work. So we hope that in the future we are able to go more regularly and observe the ones that we have relocated and see how they perform in the long run.
Thank you, Ali. Before I go on to the next question, I think this question was asked in real time while you were answering. It's a curiosity question. Was there crocodiles?
We did not see crocodiles, but recently, interestingly, there was a crocodile sighting in a nearby estuary in the area. And we know that at least one incident where a crocodile was caught in the area in the fish trap. So, yeah.
That's cool that you didn't run into any. Maybe that's why you're still here. I'm just kidding. Okay. For the next question, actually, I would like to take it from one of our viewers on YouTube. Actually, he's asking, does the depth of the sea level affect tolerance limit of corals to environmental changes? And I guess this question can be answered in general, with respect to depth, but also perhaps even relocation efforts where, like, if you transfer to a new location, does the depth matter? Perhaps Effendi would like to take this.
Wow, this is one of the good questions. Okay, I would say yes, definitely, because just imagine, okay, if you have, I'll just take the shallow reefs versus the very deep reefs where the corals are. So usually the shallower ones would have, they would say that it's always been stressed by waves, by high light, by high temperature, compared to where the deep reefs are, which are, the environment is more stable. There's not much waves, the light is relatively constant, the temperature is relatively constant. Now, if you have environmental change, a big environmental change for the shallower corals, right? The tolerance is really high for extreme environmental change because they're already used to suddenly it's very hot, suddenly it's very cold, there's a low salinity, high salinity. Now, if the deeper corals, if there's a, this is where the debate goes in, where when El Nino happens, where the seawater temperature suddenly rises up very fast, and the water column is actually now is affected by the seawater, the high temperature from the shallow down to the deeper parts of the reef. So the corals at the deeper part of the reef is usually la 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 la, they're having a good time, there's nothing changing, and suddenly there's a high temperature shift. So the deeper ones would be more affected by the higher temperature compared to the ones in the shallow. So that's how you can understand it, and that's why when you do your coral work, you do not want to breed your corals at the deeper place and suddenly outplant them to a shallower part, and vice versa. They will get a bit of environmental shock, and they will most probably we all will die. Yeah.
Okay, so we have time for one last question. I'm going to randomly take questions now. There are so many. Thank you so much, everyone. Fabian asked about microfragging. After microfragging, are they being put in tanks or back in the field? If in tanks, are you giving them additional feeding or subjecting them with any other treatments? Who would like to answer this?
Okay, I think
silence. Wow, don't everyone rush to answer it.
I'm just resting after my long spiel on depth of corals and whatnot. Okay, I'll give it a go. So microfragging. Thank you, Fabian, for this question. Just to quickly explain what microfragging is. Microfragging is to break a coral into smaller pieces and putting them near to each other, and then they will grow faster. That's the basic technology for that. So after microfragging, usually it depends, you want to put in tanks or back in the field directly. It depends on your objective of your coral rehab or your coral restoration efforts. If it's still in the tanks, yes, you need to give additional feedings because they are growing bigger and bigger and bigger. And also you have to be careful about them getting attacked by microbes and getting sick and whatnot. Yep. So you need to have maybe to put in iodine or other things to control it. That's my answer to that.
May I add?
Yeah, sure.
For microfragmentation is a technique that has been, I wouldn't say developed, but it has made a lot of noise in Florida. I was lucky to be able to partner up with someone from Florida, and that person told me that he came to Malaysia 2 years ago, and he said, well, Sebastian, everyone in Malaysia is really lucky that the conditions of the water, so in the oceans, are still good for coral growth. Because I asked him about microfragmenting, and he went ahead and said, don't think about tanks unless you've got these conditions, because one of the really last things that you want to do is actually to have tanks, what we call ex situ, coral tanks, nurseries, whatever you want to call them, because the conditions in the water are still good. He said in Florida, they are struggling not just with climate change, but sewage pollution. So microfragmenting is probably a very interesting venture to go into, but I think tanks, we have good conditions, we should make use of them.
Thank you, Effendi and Sebastian. So I know Jill said one last question, but I really want to sneak in one very last question from a viewer from Romania. She has a question for Dr. Sebastian.
Oh my God.
She wants to know, how did the passion for corals started? Because she heard about this interesting project from her teacher in Romania.
Okay, wow. Viewers from Romania, well, welcome to the show. Great to have such a far reach all the way to Europe. But let me just say I'm not a doctor. I wish I would be one, but that may come further down the line, not at this moment. Planting corals has kept us very busy. But how did the passion really come about? I guess it was exposure. I was very fortunate. My parents took me on holidays, and I remember for the first time when I saw coral reef was in Egypt, I think in 2007. And I didn't even know what I was looking at. I just saw the colors and shapes, and it was just a crazy thing to see. It was the first time I was actually scuba diving, and I didn't even know if I was breathing or if I was drowning. So it was really an overwhelming experience to see corals and to be underwater. But I do remember coming up and I said to my father, hey, do you think I can get a diving license soon? And he just shook his head and he's like, oh no, please don't tell your mother. But for me, it was exposure. Being able to see coral reefs that really ignited this interest in diving, and then diving really ignited the interest in traveling. And then one came together. I realized, well, actually, I've spent so much time outdoors, and I would love to spend more time underwater now. So what can I do to, dive as much as possible and do something? And that turned out to be corals.
Thank you, Sebastian. All right, so it's actually past 10 o'clock, which is when we were supposed to end. So again, I would like to thank all the speakers for sharing their thoughts and answering the very, very insightful and curious questions coming in from the viewers. And I would also like to thank all the viewers tuning in from Facebook Live and also YouTube Live for commenting and also asking many questions. We're very excited about the amount of reception we're getting on this event. All right, so thank you so much for spending your evening with us and joining our celebration for World Ocean Day. And I would like to just kind of put a shameless plug for all of the social media accounts involved today. Please follow Project Pulley, Coral Coup, Borneo Marine Research Station on Facebook and Instagram to learn more about our projects and receive updates on our progress. And just one last shameless plug, Project Pulley is actually premiering a short 10-minute documentary about our project. It actually was just released 2 minutes ago at 10 p.m. So if you would like to watch it, please head over to our social media and YouTube channels to watch it. Okay. And from this event, I hope that you walk away knowing much more about coral rehabilitation efforts in Malaysia and how there is not one size fits all solution. All right. And there are complexities that are still being worked out for this region. And we also hope that after this event, all parties involved, whether you're from academia or an NGO, whether you're a dive center operator or a fun diver, we should all work together to safeguard Malaysia's coral reef ecosystems for our life and the future of our oceans. Before we end, Jillian, do you have any last words to say?
Well, I'd like to say thank you everyone for joining us. And I must say that even though we really enjoyed hearing about all these restoration projects and also doing it ourselves, we have to say that restoration, rehabilitation, that's not the first choice that we want to take when it comes to coral reef conservation. We actually want our reefs to be protected. We actually don't want to be forced to restore and rehabilitate and relocate them. So, but however, we need the science for rehabilitation to be there when we actually need it. And that's why the speakers that you heard tonight are doing what they're doing so that we have a toolbox built up. But in any case, we need to protect first and prevent the destruction of our coral reefs before anything else. Thank you so much, everyone.
Thank you so much, everybody. That is all we have for tonight. Thank you for tuning in. Have a good night.
Good night.